This video will cover experiment number 12, the TLC analysis of analgesic drugs. It will go through the procedure as well as data and observations. A reference plate and sample plate were each made and analyzed. The procedure is shown on figures 1 through 7 and 10 for the reference plate and figure 8 for the sample plate. Many of the procedural steps were the same, and so pictures were not repeated. Data and observations for the reference plate are shown in figures 9 and 11 and 12 and 13 for the sample plate. The solutions used on the reference plate and sample plate are shown here and throughout other slides. To start, a TLC plate was used and a line was drawn with a pencil 1 centimeter from the bottom of the plate. On the 1 centimeter line, 6 small dash marks were made and numbered 1 through 6. Above are the 6 solutions that are used on the reference plate. Notice that a wooden pencil is used for this, not a mechanical pencil or a pen. This video is showing the spotting technique used with the reference plate. A capillary tube was used. It is a small tube that is open on both ends. It is dipped into the solution and then lightly touched onto the TLC plate where the dash mark meets that 1 centimeter line. The solvent is allowed to evaporate, and then the solution is spotted one more time to make sure that enough of the active ingredient is present on the TLC plate. Figure 4 shows the reference TLC plate after it has been spotted. Figure 5 shows the developing chamber and developing solutions that were used. The developing chamber was just a large beaker, and the developing solution is 0.5% glacial acetic acid in ethyl acetate. The developing solution was poured into the developing chamber, so only about half a centimeter deep was used. Figure 6 shows the reference TLC plate in the developing chamber. A recrystallizing dish was used as a lid. There's an arrow pointing out the current solvent level. It is about 1/3 of the way up the plate. The TLC plate was removed when the solvent was about 1 centimeter from the top. Figure 7 shows the plate after it was removed. After it was removed, a pencil was used to draw the line where the solvent reached, and then the solvent was allowed to evaporate. The same procedure was used for the sample plate. A TLC plate was obtained, a line was drawn with a pencil 1 centimeter from the bottom. These 4 analgesic drug solutions were spotted on the dash marks on that 1 centimeter line. The plate was developed in 0.5% glacial acetic acid and ethyl acetate and was removed from the developing solution when the solvent was approximately 1 centimeter from the top. In order to view the data, 2 methods were used. First, the plates were observed under UV light. This is the reference plate under UV light. Second, the TLC plates were placed in a jar containing iodine crystals under very low heat. You can see the jar in figure 10. Figure 11 shows the reference TLC plate after it was removed from the iodine jar. Figure 12 shows the sample TLC plate viewed under the UV light. And figure 13 shows the sample TLC plate after it was exposed to iodine in the iodine jar.